Friends, welcome to Biology Made Easy. Today in this video, we'll discuss some of the models of cell membrane. In the previous video, we have discussed the chemical composition. So after chemical composition, we have to discuss what is the physical structure of the cell membrane. And for the physical structure, different types of models have been proposed. Some are accepted, some are rejected. But we'll discuss all these models because they are also important from extension point of view. So the first model that was given was by unit layer. We can write here the models of cell membrane. So according to the first model that was given was by Ernest Overton. Ernest Overton, 1890. So Ernest Overton, he gave the first idea that what may be the probable structure of this cell membrane. When he gave some that is fat soluble substances to the roots, he found that the root hairs rapidly absorb the fat soluble substances. That means from this he concluded that the cell membrane consists of a layer of lipids. That means there is a single layer of, that means a cell membrane consists of a single layer of lipids. Why a single layer of lipids? Because the fat soluble substances easily pass through it. So he gave one idea that cell membrane has a lipid layer. Then let us discuss the second one. This is important. This is single layer of lipids. Single layer of lipids, this is important. Then the second theory is that of this Ernest, that is Gutter and F. Grindel. And they proposed their view in 1925. Now they took the RBC for this their experiment. Then you may ask, when we are discussing the different models of the cell membrane, why he took the membrane of RBC? It is because if this cell membrane of the cell will be taken, then the cell membrane has some extension. Sometimes it is connected with the endoplasmic reticulum. It is also modified to give rise to other types of cells. Different organelles are attached to it. But the RBC is free from all these things. That is why they prefer this RBC. That is important, why they took RBC. Now when this RBC was put in water, all the RBC cells burst. That means bursting of RBC. So when the RBC or the erythrocyte, they burst, only the membranes were separated and the liquids they came out. And the membranes which were separated, that is known as ghost. That means these are the membranes, which is known as ghost. So when the membranes are separated, then they put these membranes in water. Suppose this is the water and this is water, they put this membrane of the that is RBC, and they found that the membrane covered double the area than that is expected. Suppose they were expecting only this much, but it spread double the area of the expected. That means from this they concluded that the membrane has two layers of lipids. That is important. That means there are two layers of lipids. So according to Overton, there is a single layer of lipids, and according to the model of Gritter and Grindel, there are two layers of lipids. Then after these two, some other models were given. They are known as trilaminar models. Trilaminar models. So in this trilaminar models, there are three layers. And according to this trilaminar model, when the cell membrane was observed through electron microscope, it was found that it looked like a railway track. It looked like a railway track. That means it looked like a railway track, and inner side, this is cytoplasm. This is the inner side. This is the cytoplasm. And it looked like a railway track. That means the outer two railway tracks, they are quite opaque, and these are the protein layers, proteins. While the middle one, which is a transparent, this is the transparent zone, transparent, and it consists of lipids. That is why it is known as trilaminar. Though in the lipids there are two layers, but this is they consider it as one, and on either side there are two protein layers. That is, again, this is very important for examination point of view. This is very, very important. That means in the cell membrane, the two protein layers are present. They are opaque, while the middle layer is transparent, and that consists of the lipids. So this model, or this type of structure is known as trilaminar model. Under this trilaminar model, again, three different types of models are there. That is the model of Daniel and Johnson, which is known as sandwich model. Then the model of Robertson, known as unit membrane model. And the third one is fluid mosaic model given by Singer-Nicholson. So we'll discuss one by one. So here the first model is that of Danieli and Dawson. Danieli and Dawson model. So this is the Danieli-Dawson model was given in 1935. So according to this Danieli-Dawson model, there is a bilayer of phospholipids. There is a bilayer of phospholipids, and on either side of these phospholipids there are proteins, but these proteins are hydrophilic, and they are known as alpha globular proteins. That means these are the two layers of these are the lipids, and on either side there is there are proteins, and these proteins, they are surrounded by water. Like this. So these are the proteins, and these are known as alpha globular proteins. Alpha globular proteins. And again, these alpha globular proteins are attached with this bilayer of phospholipids by means of electrostatic forces. That means they are joined together by means of electrostatic process. Electrostatic process. Now, the bilayer of phospholipids, they are arranged in such a way that this is the head of the phospholipid, which is polar, or it is hydrophilic, and this is the tail. The tail is non-polar, or it is hydrophobic. Now the two layers of phospholipids, they are arranged in such a way that their tails or the non-polar hydrophobics are present towards each other. And how they are connected? They are connected by means of hydrophobic bonds and van der Waals forces. That means these, they are connected. That means the non-polar ends are connected by hydrophobic bonds and van der Waals forces. Now these are very important for examination point level. And again another important thing. Now the non-polar are present towards each other and toward the interior side. That means they are completely away from the aqueous solution present on either side. This is also important. That means the polar heads are present toward the inner side and outer side, while the non-polars are present toward the inner side. So this is the structure that is given by Daniel Dawson. That is trilaminar model, because on the two sides the proteins are present, and here one lipid bilayer is there present, and the proteins are alpha globular proteins, and they are hydrophilic, and the hydrophilic and proteins, they are associated by means of electrostatic forces, while the non-polar tails, they are connected on either side. They are connected by connected by hydrophobic bonds and van der Waals forces. This is the structure given by that of the Daniel and Dawson, and this model is also known as sandwich model. Now why it is known as sandwich model? Because the lipid bilayer is sandwiched on either side by two layers of proteins. The lipid bilayer is sandwiched on either side by means of proteins. That is why it is also known as sandwich model. Then the next model is, will give that of the that is Robertson, and this Robertson model, that is Robertson gave the model in 1959, and his model is known as also unit membrane model. His model is known as unit membrane model. Now we should first know why he gave the name unit membrane model. When he studied the structure of the cell membrane, he found what is the structure. We will discuss. But again he found the same structure is present in the nuclear membrane and in all the membranes of the organelles, mainly plastids, mitochondria, endoplasmic reticulum. That means all the biomembranes have similar structure. That is why he gave the name unit membrane model. Now what is his model? Now his model is to some extent similar to that of the Daniel Dawson model, sandwich model, with certain modifications. So what are the modifications we have to see? So according to him, there is also a bilayer of phospholipids are there. Now the bilayer of phospholipids, this is about the inner side, this is the outer side. In the outer side, the proteins which are present, they are different from that of the inner side. This is the basic difference between these two models. That means the proteins which are present on the outer side, they are like, suppose this is the protein which is present, and this protein is known as a mucoid protein. And when we call a protein mucoid, when the protein is associated with the carbohydrates, that is mucoprotein. But those which are present on the inner side, like this. This protein is non-mucoid protein. So non-mucoid protein means it is not associated with any carbohydrate, while the outer side is associated with the mucoid. This is the basic difference between the model of Daniel-Dawson sandwich model and the unit membrane of Robertson. Then again he studied that the protein layers, their width is 20 angstrom on either side, and that of this bilayer phospholipids is 35 angstrom. That means the entire thickness of this cell membrane is 75 angstrom. But sometimes from membrane to membrane it varies. So sometimes it is written that it varies between 50 to 100 angstrom, because in some cases it varies from model from one to another. Now these are the two models from the trilaminar model, but both two models we have discussed. We have not discussed the third one. Now these two models under this trilaminar model, they are rejected at present. This is proposed in 1959, then in 1935. For some time they continued. Till 1972, both these models continued. Particularly this Robertson model unity membrane, they continued till 70s. But after that it was rejected. Now why these two models were rejected? So what is the reason? Now we can write here the reasons for rejection. Rejected. Why they are rejected? Because they do not explain the permeability. That means how they say different types of ions and electrolytes, how they move, they do not explain. Then the second point, these two models, they provide that the cell membrane is stable. That is, cell membrane is stable. So when the cell membrane is stable, it doesn't explain all the functionality of the cell membrane. That is the cell growth, cell reproduction, secretion, how all these things will take place, because this is stable. So since it is stable, do not explain, both the models do not explain the functionality. Then there is a third point. Now, already we have discussed in the chemical composition that the lipid protein, the lipid is to protein is usually 40 to 60 usually, but according to these models, the lipid protein composition is completely different. So these are some of the points. That means according to these two models, they are stable structures. So what are the different, how the different types of functions like growth, reproduction, secretion take place, they do not explain. The lipid protein ratio, that is 40-60, that is also not possible according to this. Then that is they do not explain regarding the permeability of the membrane. So that is why these two are rejected. Now the next model, third model, is the accepted model, and that is known as the fluid mosaic model given by Singer and Nicholson. That we'll discuss in the next video. So thank you very much. Wish you a very bright future. God bless you.